So now we will discuss the microscopic view of the retina as well as the identification point of the histology slide of the retina. Before we see the actual slide of the retina under the microscope, we will discuss the schematic diagram of the layers of the retina. So this is there are total 10 layers of the retina. This is the number one layer, that is the pigmented cell layer, which is containing the pigment granules, and that is of the low cuboidal type of the cells. After this pigmented cell layer, another second layer that is situated here. In this there is a presence of the rods as well as the cones, that is the photoreceptors. After that, the third layer that is shown here, this is representing the external limiting membrane, and that is a sieve like structure which is pierced by the segments of the rods as well as the cones. And this external limiting membrane, that is formed by this molar cell, particularly the outer process of the molar cell, is going to unite with each other and forming this external limiting membrane. After that external limiting membrane, you will find this nuclear layer, which is containing the nuclei of the rods and the cones, and that is known as the outer nuclear layer. After that, there is a outer plexiform layer, which is containing the synapse between this processes of the photoreceptors as well as this bipolar cell, and this is the horizontal cell. So the processes of all these cells, they are combining and forming a network, and that is lying in the outer plexiform layer. After that layer, the sixth layer is containing the again the nuclei of the bipolar cell, that is shown here in this brown color, that is the inner nuclear layer. After that, again there is a plexiform, inner plexiform layer, but here the plexiform layer is formed by the processes of the amacrine cell with the bipolar cell as well as the ganglionic cell. After that inner plexiform, again there is a nuclear layer that is containing the nuclei of the ganglion cells, and that is the ganglionic layer. Now the processes, particularly the axons of this ganglion cells, they are combining with each other and lying in this layer that is known as the nerve fiber layer, and that is going up to the optic disc to form the optic nerve. And the last one, that is the most inner layer, that is the inner limiting membrane, and that is formed by this again molar cell, particularly the inner cell process of the molar cell, that is forming the inner limiting membrane. So this is something regarding the schematic diagram of the various layers of the retina. Now when you see the actual microscopic slide of the retina, you have to remember that this first layer, pigmented cell layer, is not seen in that slide. Okay, so never speak the pigmented cell layer in a viva when you are examining the slide and giving the viva, because this pigmented cell layer is firmly attached with the choriocapillary layer of the choroid by the Brooks membrane. So whenever there is a preparation of the histological slide of the retina, this is going to be detached with the remaining 9 layers of the retina and remain attached with the choriocapillary layer of the choroid. So you will not find in the slide of the retina. Now this is the actually slide of the retina under the 40x magnification. So in this you cannot see the pigmented cell layer, which is the first layer of the retina. So after that you can see the layer number 2, that is representing here this A with the red arrow indicating this is the layer of the rods and cones. After that you will find the number 3 layer, external limiting membrane, that is showing here, you can see with the yellow arrow, and that is lying between the layer of rods and cones as well as this nuclear layer, outer nuclear layer, which is containing the nuclei of the rods and cones, that is shown in this sky blue color arrow. So this is the number 4 layer, outer nuclear layer. After that, there is the plexiform layer shown in the light green arrow, that is the outer plexiform layer. After that, the blue arrow is indicating again the nuclear layer, that is having the four types of the nuclei of the four different types of the cells, that is the inner nuclear layer. After that, again with the purple color arrow, this much area that is representing the inner plexiform layer. After the inner plexiform, number 8, again you can see the nuclei in this layer, that is representing the nuclei of the ganglionic cells, so that is the ganglionic layer. After that, brown arrow is showing here, this is again the nerve fiber layer. Okay, the fibers emerging from the ganglionic cell layer. And last one with this gray color arrow, the edge of this microscopic field. Okay, you can see the edge that is representing the inner limiting membrane. So that's all about the layers of the retina under the 40x magnification. So you have to remember that the pigmented layer is not present when you see the slide of the retina under the microscope. Thanks a lot.